Hi everybody, I'm Steve Glavas. I work for the Swedish Institute of Communicable Disease Control. It's a mouthful, it's a big, long name. And my division is called the Genomics Division. So what we generally do is we try and implement new methods for infectious disease control. And we speak with our different expert groups. And in this particular case, we work with our influenza group. And what they wanted to do was develop something that was good for pandemics. And that would be Ion in this particular case. So this is our institute. And there's the name of it, Smittskydds Institute. There's a lot of consonants in there. But we're based in Stockholm, Sweden, right on the Karolinska campus, right across from Science for Life. And what we do is we work with vaccines, we do infectious disease control, we fight outbreaks. And the research we do isn't really research, it's more methods development. So we're trying to make sure we have the best techniques available for doing infectious disease control. And the project we worked on was influenza, so I figured I'd tell you, what is influenza? It's a very tiny little bug, it's a virus. It's got 8 genome segments. And generally what people do is they look on just 2 of these today. That's H and N. Those are the most common gene segments that you're looking at. But everyone's probably heard about the pandemic from 2009. There were about 14,000 deaths, according to the ECDC, that's the European Centers for Disease Control. And then there were, what, 43 to 89 million cases in the US. So that's a lot of samples that people need to get through. They need to be able to screen and find out if it is something that will cause a pandemic and will cause a lot of deaths. So we need to keep track of that. But what we did, we joined a network. This is also facilitated with LifeTech. It was very nice of them to put us all in touch. I'm in the top right. And then we also worked with JCVI, the CDC, USDA, and also our friends in Italy, the ISS in Rome. And everybody put in a little bit of effort into it to find out what they could do with this influenza network. And we started producing some data. But I wanted to give you a little background of how we do it today. A lot of you already are familiar with next-gen techniques, and you start seeing these wonderful things like Sanger sequencing is antiquated. But for us in the clinical world, we have to have these tools, which we have very well understood over 10, 20 years. So we have very slow processes that work very robustly and handle everything perfectly. And a 3-day Sanger assay, it's great. Gives us the data we need. It does everything we need it to do. It's just we can't scale up in case of a pandemic. It's impossible. In fact, I'll show you some slides on what we tried to do in this little experiment a little bit later. So the NGS method is basically a whole genome amplification, all 12 kb of it, and put that onto a library builder. That's one of the newer pieces of equipment that we have. It's a small automation device for prepping either RNA or DNA extraction as well as creating libraries. And of course, we put it into our Ion machine. And then we also have an automated pipeline, which is actually kind of key for the whole thing, that we can analyze everything and come out with those CLADE reports that we require for feeding back into the international databases for pandemic control. But the whole genome amplification kit, this is a gel. Sorry, guys. I don't like gels myself, but a lot of people in our network are wondering, this gel image, different bands, some are strong, some are weak. They're different lengths. How robust is this assay? How is this going to look in next-gen? Can we actually afford to go on with this project? So we're used to running everything on a gel to begin with. But what we found was that this gel intensity was really not critical. And I'm sure many of you already seen that, the ones that have used NGS before, that you're just capturing a lot of fragments, you're doing a lot of sequencing, and it doesn't really matter how much coverage you have as long as it's roughly equivalent. And we didn't have the biggest budget. So all these wonderful machines you see on this slide, we didn't have any. So we just had this one, and this is all we had to go with. We ended up getting something like a bioanalyzer later, And we actually saved some of the libraries to go back and quality control them and start comparing some things that we did between automated prep on the library builder versus manual prep on eGels. And we found that the manual prep, obviously, you're doing a lot of hit and miss and you're pulling it out at the wrong time, but you get nice tight peaks. But that actually affects the data in the sense that your libraries are much too small and you don't get the long reads that you really want. Whereas the library builder, you'll see in this presentation, it really didn't affect the data that much. And there's a library builder peak. You can't really see a peak. It looks really broad. It doesn't look very good. But we had all these other peaks for the manual method. But we didn't use a manual method in this particular case. So here's our workflow. We had an antique robot. It's been end of life by the manufacturer for quite some time. And they're telling us we have 5 years left. And OK, well, we got to start looking for something else. So we did our imaging on a gel. We did everything on the library builder that we could. And we hope for the best. So that's what we did. This is methods development right now. So we aren't actually using patient samples here, just to calm everyone down. But then we changed our whole workflow to this. This is what we're going to at this point. We ended up buying an automated instrument. It's called a LabChip GX. It's equivalent to the bioanalyzer, but it's a 384 well plate that you just load everything and you go away. And it's compatible with the LIMS system, which is what we need for our workflow. So we can start reporting everything into a central database and no more gel images, which is wonderful in my book. But this pipeline that we have today runs about 26 samples. And 26 influenza genomes per day, that's a whole lot. You usually don't do that many. In a whole season, we're looking at maybe 400 genomes that we might be looking at. So that's just a couple of days worth of work, and we got it all covered. But once we start moving the entire workflow over to the NGS platform, we'll probably need some bigger robots to do the job for us, just because the library prep is definitely the bottleneck for us now. Our experimental design, we had a new hire. He started on a Monday. On Tuesday, I said, here you go, let's get going. He got his phone, he got his computer, and then he logged into our little electronic laboratory notebook system and said, OK, this is what I have to do. We gave him 20 isolates of influenza A. And we have a Fluent kit that we were given by Life Technologies to use in our network. And we tested that, a 10-plex on each of the chips, a 314 and a 316. And we wanted to check those against some samples that clinical samples, not isolates, that actually had Sanger sequencing done on them, just on the H and N genes, because we don't do whole genome sequencing with Sanger. And then we wanted to actually validate the system, technically validate, see if there are any differences between Ion torrent sequencing versus Sanger sequencing, and what do we see in this with our samples in our hands. So we went on. And we got some data. And this is a customized report that we've made at SMI. And this is just basically what you would see on your PGM report, read length and read distribution and quality values. And then we also have the different fragments. So these are the 8 gene fragments here. And you see them just from top to bottom. And they're very even coverage. These are amplicons. And we don't really have N bias, which is pretty unheard of when you're looking at all the different samples I've run with NGS and doing any kind of amplicon sequencing. You always have to do some kind of N depletion, and we didn't really have to do it here. We just took the sample right out of the plate, put it right into the library builder, right into Ion torrent, and got this wonderful data. And this is with our wonderful pipeline with none of those magic tools that everyone seems to have in their lab that we don't. And then we create the report that we need to upload and make a decision, an actionable report. So this is the information that we give to our experts for influenza and say, here, you can now find these mutations on this particular genome, make a decision, what do you need to say to the patient? And it's in place. We used an assembly algorithm called CLC, but to give you an idea of what speed it is, we have a very specialized compute cluster for this. It generates assemblies for these 12K genomes about 6 seconds per 314 sample, and about 47 seconds for a 316 sample. So there's more data from a 316 chip per sample in this particular case. But this compute cluster, it's our bioinformatician's laptop. Works pretty well. We're hoping to get something more professional than this right now. And to give you an idea of what the coverage we had, for the different genes, we have the gene segments on the left. We have the length in the second column. So you can see they vary from about 1,000 bases to about 2,500 bases. And then you see how many reads we have and the average coverage per base. So on the 314 chip, you get anywhere from about 50x coverage, which is good enough for us, up to 710, which is way overkill on the different fragments. And then you see that it'd be a 10x multiplier when you go up to the 316 chip. So you see roughly 10 times the data. So it worked pretty linearly for us. And then we had to validate our old Sanger data versus the isolate data that we had. And we started finding some differences. The 3 different genes, they're H, N, and M. So in the 2 blue columns on the left, that's the H gene. And there's 2 columns in that blue region. The 1 column is the Sanger data that we had from previous on clinical patient samples. And then the second column is what we had from our Ion torrent. We were comparing them and finding out what the differences are. And if you see red, that means that there was a difference from the 2 data points. So we started wondering what the heck just happened. How can we have a difference in the sequencing between these different samples? So we found something. And in this particular case, it's the Stockholm strain. It's a good one to pick. But we had 3,200x coverage at this particular position. So we knew it wasn't a sequencing error. And we're wondering what could it have been. And obviously it was just because it was an isolate, not a clinical sample. So there's a lot of selection going on in these particular genes when we actually culture them. So what we were seeing was biology. But I wanted to be sure, so we started a campaign of doing the whole genome sequencing. But that was 106 oligos that we purchased, separate primers. And there was a very nice service provider who's actually here and ran a lot of these samples for us. And there's just not enough time to do it all. And in about 3 weeks' time, we just couldn't get through them all. It's only 12K. It should work fine, but you have to go through and optimize all these different samples and different primer concentrations and how they behave. But when we designed the whole 106 samples, 106 primers, we actually used something in-house, a SME primer. It's something that we use for watching primers and how genomes evolve over time, and make sure that our primer systems actually work for whatever disease-causing bug might actually be out there, and make sure it does work. I don't want you to see any details on here, except that if you see a digit in one of these columns, that means there are 0 differences. If you see no digit, it means that we didn't get to it. So every single one checked out. We had no issues with Sanger whatsoever. So it was a perfect comparison, Ion torrent versus Sanger on this particular experiment. So some fun facts. Our entire system error for the NGS workflow that includes extraction all the way through to analysis, we had a 1% error rate for our NGS workflow. Longest read, my bioinformatician, he gets a kick out of me when I say, all right, what was the longest read? He's like, it's not important. I know, but we got to put it up there. We got 384 bases, yay. And the highest coverage for any given base is 966 and 12,696x. Feels good. And this is my thank you slide. I wanted to say thank you to Jonathan Rothberg for his uncanny ability to take 2 totally incongruous things and make a wonderful package like Ion torrent. Like my tie? But really, the big thanks is for SMI, genomics platform, the Influenza group. Science for Life lab in Uppsala, as well as life tech support. And of course, I want to give a special thanks to Sweden Order for helping us get all our products on time in a timely manner. And of course, there's going to be a networking party this evening. I was told to show this slide. Hmm? Yeah.